continue our presentation on synthetic biology, biotechnology, bioengineering, we talked about biological clock, innate mechanism that controls the physiological activities in an organism that change on a daily, seasonal, yearly, or other regular cycle. The video link below shows a video where scientists at the University of Georgia are studying mold on bread to see their biological clock and trying to understand when's the best time for medication to be given, to help with cancer research, because every living organism has some form of a biological clock. And how can we, as scientists and engineers, hack that? To, especially, like, for example, they discussed in this video cancer and trying to fight cancer. Three major questions that go along with the biological clock: how does it work, how does it synchronize, and how does it translate to the activity? We have a few examples of day fly, etc., and how time is a major important factor with this. All right, review of what we covered previous week. We're going to look at DNA. Here's a quote by Francis Collins. The building blocks of DNA, right here. Every cell in our body has the same DNA, no matter what type of cell it is, but there's slight chain differences. We have genomes equal 46 chromosomes, 23 pairs, approximately 3 billion base pairs. And the thing that's crazy here is that we are 99.9% identical. Only 0.1% is unique. All right, what makes one cell different from another? And we look at the alleles in the gene segment of DNA. It tells the cell how to make the certain proteins. That part is very important. Now, because of gene sequencing, here's an example of a normal hemoglobin protein versus a mutant protein such as sickle cell. One area is different, okay, right there. Then we looked at the Human Genome Project. The goal was to sequence the human genome. This was an international project done all over the world. They took DNA samples and scientists sequenced DNA here. The human genome is a complete set of genetic information for humans. Okay. Below are two links that dig in deep into the human genome and will give us more information on that. You can go through those links. About the first link above is an interactive timeline just about genetics as a whole and the evolution of it over the course of the last 100 and some odd years. And then below is a interactive animation on the Human Genome Project and DNA sequencing and more. Okay. This was a sequencing DNA game that we used here. Then we started looking at forensics. And the reason we're looking at forensics is engineers build tools for forensic scientists, as well as specifically on the medical side of forensics, what we're looking at. We watched two videos here about forensics after going over the definition of forensis, a relevant modern definition of forensics. It's any science used for the purpose of the law. Now, forensic DNA analysis relies on two basic factors. Every individual, except identical twins, has unique DNA sequence. Each cell in an individual contains the same DNA. That could be skin or that could be white blood cells, still have the same DNA. DNA has been used to solve crimes, identify human remains, missing persons, determine relationships between family members, etc. Using DNA evidence to help solve a crime, they can collect DNA. They can determine if a person is that person or not based off of their individual specific DNA. And they can collect DNA from any human fluid, skin cells, etc. They can find that on a variety of things. Now, all right. DNA sequence of any two people are 99.9% identical, and here's an example of this DNA sequence. Just one letter is different in this genome here. And forensic DNA analysis focuses on the difference between individuals, and they call these variable regions. And here's an example of a variable region of short tandem repeats. Scientists look for these to find that individual marker. And STRs can have different numbers of repeats, as seen here. Polymer chain reactions. PCRs allow you to make many copies of a target sequence. Okay? In this next couple slides of the process of this. Okay. Now the PCR is primarily attached DNA sequence on either side of the repeats. These regions do not vary between individuals. The same primaries can amplify the repeat because they are the same. All right. There's more to this process. All right, a little bit more about this. We know that everyone inherits 2 copies of each STR, one from the mom and one from the dad. Okay. Now, DNA profile. Main reason we're looking at this is that the combined DNA index system is a collection of DNA profiles, and they help use to find missing persons as well as solve criminal cases. And they've been using this since the mid to late 80s. All right. And it's because of everyone having their own individual DNA. It also helps identify relatives as well. And you can see it's been being used to solve crimes. Look how many it's been done since 2009. More on that to help solve crimes. All right. And it's helping expedite crime cases where they can eliminate suspects because of DNA. Also, this has to be handled with care because you're dealing with biology, living organisms, living things here. They can be contaminated, bacteria, mold can grow, etc. So there has to be standards that are set and put in place to ensure that this is done with a quality control aspect. Evidence can help overturn convictions. This has been done starting in the late 80s, where people have been released from prison, jail, who were wrongly convicted because at the time when their case went through the process, there wasn't this science out there yet, this engineering. And then they store in evidence lockers so that people can go back and look at the old evidence, in this case DNA. And we get more techniques. You can get DNA profile information from much smaller samples and older methods. All right. DNA analysis help find recover missing people, especially with such as Hurricane Katrina. And then we play these forensic games. Okay. We only got to the first one. Now, thank you and take care.